Good afternoon, students. So we were doing biomolecules chapter, and in that we have studied the introduction part. That biomolecules are the all the organic compounds which are produced from and found in the cell. If we fractionate a cell, if we break the cell, and then we whatever organic molecules or hydrocarbons that we get from the cell, that are called as biomolecules. So biomolecules are all the organic molecules which we get from the cell. So these are the diagram. These are the diagram of the biomolecules as it is given over here. This is the image of the biomolecules. And this is chapter 9, class 11th biology. Sugars, we can see two sugars are shown over here. One is hexose sugar, that is glucose, and other is pentose sugar, 5 carbon, one carbon here, one carbon here, one, two, three, four, and fifth carbon is in the chain only. So we say that it is a 5 carbon sugar. So ribose, name is given ribose. So these are the sugar units, and we also can say these are monomeric sugars. Okay, so monomer sugars mean single unit sugars. These are when they come in the chain form, then they become carbohydrates. Amino acids are shown over here. There are three amino acids which are shown. Glycine, this is simplest amino acid, not having any alkyl group. Instead of alkyl group, it is having hydrogen only. Second is alanine. Alanine is having methyl group over here, alkyl group at the R position. So in each amino acid, the R changes. Alkyl group changes. In alanine, it is CH3. In the serine, you can see it is CH2OH, hydroxyl, hydroxymethyl. Okay, it is hydroxymethyl in the serine amino acid. So this is the we can say different types of amino acid on the basis of different R groups present in them. All other groups are same. There is carboxylic group, there is amino group, one is hydrogen, other which will be changing will be R group. R group will be continuously changing. Now coming to the next, palmitic acid, palmitic fatty acid. This is 16 carbon, you can see over here, a long hydrocarbon chain fused with a carboxylic group. So this is your palmitic acid. Then glycerol, you can see glycerol is shown, it is propane triol, and 3 carbon are attached to 3 hydroxyl groups. So this is glycerol. When each hydroxyl group of glycerol is fused with 3 different fatty acids as shown here, we can get the triglyceride. We can get triglyceride where R1, R2, R3 are 3 different fatty acid hydrocarbons. R1, R2, R3. So we can get triglyceride in this form. Now, one other triglyceride is there, which is phosphoglyceride, because phosphate group is added to one of the hydroxyl groups. Two are the fatty acids, and the third one is fused with the phosphate group with a nitrogen also, nitrogen compound also as well as. So this is phospholipid lecithin. Then cholesterol is shown, which is a we can say multi aromatic, multi-chain aromatic compound. You can see it in the form of multi-chain aromatic compound. So this is cholesterol. So now we are going to understand these things again. Are you able to see this video? Tell me, are you able to see this video?
Proteins are chains of amino acids linked by peptide bonds. The 20
Yes,
different
ma'am.
amino acids used to make all proteins differ only in their side chains, and the properties of these side chains account for the great diversity of protein structure and function. 10 of the side chains are
So proteins are joined by the peptide bonds. It was shown in the previous, it was shown like this. The peptide bond is shown over here as yellow mark, and it is the carboxylic group of one amino acid of N terminus and amino group of the C terminus amino acid. When two amino acid fuses, then we get a peptide bond. Okay, so two amino acid are fusing, and here one bond is formed which is between the C and end. So carbon and nitrogen are joined with a peptide bond, and this bond is known as peptide bond, which is between the two amino acids. So here hydrophilic side chains, this is the amino acid side chain R group. It is hydrophilic because it is having hydroxyl group. It can bond hydrogen bond, it can make hydrogen bond. Here hydrophobic side chain because only methyl group is there. So on the basis of the side chains, the amino acid can be classified whether it is hydrophilic if it is having any group which can make bond with water, and hydrophobic it cannot react with water. So these things are there in the first in this video. One is peptide bond, second is hydrophilic side chain, hydrophobic side chain. Now they are moving towards the different types of amino acid on the basis of side chains, whether they are positive amino acid, negative amino acid, basic, acidic, what
used to
are
make
the
all
types.
proteins differ only in their side chains, and the properties of these side chains account for the great diversity of protein structure and function. 10 of the side chains are hydrophilic, either polar and uncharged, or electrically charged.
So lower side, you can see there are positive or negative side chains. Okay, aspartic acid is having COOH negative group. Glutamic acid is also having CH2 COOH. Lysines, on the other hand, histidine and arginine are having positively charged groups. On above, you can see uncharged polar groups. Okay.
7 of the side chains are hydrophobic. 3 of the side chains fall into a special case category based on their structural features.
These three are special case um, amino acid. Why? Because the this is cysteine is having sulfur in it, sulfur containing amino acid. This is cysteine. Glycine is what? It is having H only in the form of side chain. I told you it is simplest, and it is having H in the place of R. And proline is having the interconnected, we can say, side chain. A side chain which is coming from carbon and connected to amino group only. So it is the interconnected type of amino acid. Clear?
Although glycine and proline generally are hydrophobic. Collagen is an example of how a protein's amino acid sequence determines its.
So here collagen protein structure is explained. It will help you to understand that how the amino acid structures determine the protein structure. This is the polymer of amino acid. Collagen is the polymer of amino acid. It's having many amino acids, and it is a triple helix. Means three chains, green color, red color, and the blue color chains are intertwined with each other to make one protein, which is known as collagen protein, and it is most abundant protein found in your skin and your hair.
Structure and function. Amino acid sequences are encoded in the DNA of genes. Collagen is the most abundant mammalian protein. It's the main component of skin, bones, and teeth. This protein is composed of three helical polypeptide chains that form a stiff, supercoiled cable. The type of helix in each polypeptide is unique to collagen and results from collagen's unusual amino acid composition. A collagen helix forms largely from the influences of two types of amino acids: proline, which introduces sharp twists in the polypeptide, and glycine, which has a small side chain consisting of hydrogen that doesn't interfere with packing in the helix. Collagen contains many of these amino acids, but few bulky ones, for example, phenylalanine. Each helix contains three amino acids per turn, with glycine located at every third position. The three polypeptides in a collagen molecule associate with their glycines all facing collagen's center. Glycine is the only amino acid small enough to allow the polypeptides to pack by hydrogen bonding into a tight cable. Individual collagen molecules cross-link to other collagen molecules to form tough collagen fibrils. Collagen, from which gelatin is derived, is a poor source of essential amino acids, the amino acids that the body cannot manufacture and must receive from the diet.
So, some of the amino acids, as it is shown here, cannot be synthesized by body. They are only derived from the source, or they can say from the food source, meal, what we get. So there are two categories of amino acid on the basis of the from where we get it, and the amino acid which are synthesized from in the body are known as non-essential because they can be made inside the body through the reactions, through the chemical breakdown of what we can say one amino acid can change into another. Through there are chemical reactions which make them. So inside the body they are synthesized. They are non-essential. But those amino acids which are can only be obtained from food, can only be obtained from external source, those amino acids are known as essential because they are not synthesized in body but they can only be derived from the food that we eat. Okay, so there are two kinds of amino acid in the basis of their synthesis or need in the body, how they can be used in the body: essential and non-essential. Those of which are used from the or we can which we get from food are essential, and those which we do not need from food and we synthesize in our body are non-essential. There are 8 amino acids which are essential out of 20 naturally occurring amino acid. Out of them 8 are essential. Okay, you can search the name on the net that what are the names of those 8 essential amino acid. Now carbohydrates.
Carbohydrates include simple sugars called monosaccharides, as well as large polymers called polysaccharides. Glucose is a hexose, a sugar composed of 6 carbon atoms, usually.
So the glucose is a monosaccharide, a 6 carbon molecule in which it is arranged in the ring form, and it is monosaccharide because it is single sugar unit. Saccharide word means sugar, and starch is a polysaccharide because it has many glucose attached with bonded with each other to form a long chain. So it is a polymer of sugar. So it is called as polysaccharide. Poly word is always we can say prefixed in front to know that to mean it means that the thing is a polymer, polysaccharide. Like for example, we say polythene. So polythene is what? The polythene is a polymer of the ethene, and we use it as plastic. So in the same way, the polysaccharide is a polymer of the glucose. Starch is a polymer of glucose in which large number of glucose are joined or bonded with each other to form starch.
Found in ring form. A starch macromolecule is a polysaccharide composed of thousands of glucose units. Glucose molecules can be added to starch by a condensation reaction. In condensation reactions, two molecules covalently bond to each other and release a water molecule. Here, the bond...
So this is important, that how we can make starch from the glucose. So through condensation reaction in which a covalent bond is formed and a hydrolyze, we can say water is released, dehydration occurs. So when one of the glucose molecule at fourth position fuses its hydroxyl with the first position of the hydroxyl, then we can say two hydroxyl groups will be nearby each other, and a water molecule will be released from the between, and both the sugar gets attached. So this simple reaction is known as condensation reaction. In the biomolecules, whether the formation of polypeptide bond means the bond between two amino acid, or whether the formation of nucleotide, we will study it in the next topic, nucleic acid. So there also, all everywhere, wherever the monomers are joining, everywhere the water is released and the condensation reaction is taking place. So here the condensation reaction has resulted in a bond which will be known as the glycosidic linkage. This linkage is known as between the fourth position of one of the glucose and the first position of one of the glucose, between 1 dash and 4 dash position. This is 1 dash 4 dash alpha glycosidic linkage, because alpha glucose are involved. So alpha glycosidic linkage
forms between the first carbon of one glucose and the fourth of the other, creating an alpha 1 4 glycosidic linkage. Branching of new chains can also occur between carbons 1 and 6. Different types of starches are...
So carbon 1 and 6, if from one of the sugar, and at the sixth position a new sugar is attached with the first position, so here also same way the water molecule is released. So this is the branching, how the branches are formed in the linear chain. If the linear chain is going on 1,4 glycosidic linkage, and somewhere if another chain get attached at the sixth position, then this becomes the branched polymer, and this linkage will be called as alpha 1,6 glycosidic linkage.
In fact, distinguished by the amount of branching. Amylose, or plant starch, is not highly branched. Glycogen, by comparison, is highly branched. This polysaccharide is stored in animal...
So amylose, that is the plant starch, is not highly branched, but glycogen, that is stored, if we eat large amount of carbohydrates, we store it in our liver, and liver store it in the form of glycogen, that is a highly branched polysaccharide.
Livers and muscles.
Muscles also store it.
Polysaccharides are forms of stored energy that can be easily hydrolyzed to yield glucose. Glucose can then be further broken down to release energy that's used.
So carbohydrate is a source of large amount of energy. We say that in our meal we get to 60% to 70% our food is carbohydrate. So they are energy rich food. They can release the energy by fast, we can say quickly, by getting digested. So when they are broken down, the energy present in their bonds is released. So they are high energy molecules. And when they are released, firstly they are broken down in glucose, and the glucose is also broken down in mitochondria during respiration, okay, to release energy in the form of ATP, that is used for muscle movement, that is used by the organism for various kind of activities.
In cellular activity.
Now lipids.
A triglyceride, also called a triacylglycerol, is composed of 3 fatty acid molecules and 1 glycerol molecule.
So you can see very well here that in the top, which is marked with the blue color, that is a glycerol component, that means propane triol, and then there is a ester bond, ester functional bond of the hydrocarbon, And here the OH of the glycerol is fused with the OH of the carboxylic group, and water is released again, and the fatty acid chain is fused with the glycerol. And this structure where three fatty acids are fused with three OH of the glycerol, it becomes a triglyceride. Is it clear? The same is given in your NCRT as well.
Yes, ma'am.
Fatty acids attach to the glycerol molecule by a covalent ester bond. The long hydrocarbon chain of each fatty acid makes the triglyceride molecule nonpolar and hydrophobic. Palmitic acid is a fatty acid with 16 carbon atoms. It's called a saturated fatty acid because all the carbon atoms in the chain are single bonded to each other and are fully saturated with hydrogen atoms. Fatty acids, such as palmitic acid, are built by adding 2 carbon atoms at a time to smaller fatty acid molecules. A cell can use palmitic acid to form other fatty acids, such as oleic acid. In this process, oleic acid is formed by adding 2 carbon atoms to palmitic acid, and then by inserting a double bond between carbons 9 and 10. Because oleic acid has one double bond, it's considered a monounsaturated fatty acid.
So saturated fatty acid will have all single bonds and will be straight linear lipid. But when a double bond will occur in the chain, so that will become a kink is there. Kink means twist will be there in the long hydrocarbon chain. And if one time only there is double bond, it will become monounsaturated fatty acids. यानी MUFA, MUFA, monounsaturated fatty acid. But some are polyunsaturated fatty acids. We will see it now. They are having 2 or 3 double bonds in their chain. So they are extra twisted, and they are polyunsaturated fatty acid PUFA, PUFA.
Polyunsaturated fatty acids, such as linoleic acid, have 2 or more double bonds. The double bonds kink these molecules and prevent them from packing tightly together. The loose packing results in triglycerides that are liquid at room temperature. Mammals cannot make linoleic acid. It's required in the diet. A glycerol molecule makes up the backbone of a triglyceride.
Plus,
Make linoleic
this
acid.
linoleic acid, which is found in the oil, seeds, nuts, safflower oil, these are rich in these are PUFAs, and these are very good fats which decrease the incidence of arteriosclerosis. That means heart diseases. So they decrease the chances of a person who are eating these fruit, nuts, and seed oils. They are they are they are having less chances of getting cholesterol in their arteries. This is a good thing for them.
Required in the diet. A glycerol molecule makes up the backbone of a triglyceride, in which glycerol's 3-hydroxyl or OH groups form ester covalent bonds with 3 fatty acid molecules. The covalent bonds form by condensation reactions in which water is a byproduct. Triglycerides can contain a mix of saturated, monounsaturated, and polyunsaturated fatty acids. They provide a concentrated store of energy. DNA and RNA are nucleic acids, polymers of nucleotides. 2 polymers with complementary nucleotide sequences can pair with each other. This pairing endows nucleic acids with the ability to store, transmit, and retrieve genetic information. 2 strands of DNA pair by hydrogen bonding. A component of one nucleotide called a base forms a hydrogen bond with a complementary base on the opposite strand. The base cytosine, C, pairs with guanine, G, and adenine, A, pairs with thymine, T. The monomer of the nucleic acid, the nucleotide, is composed of 3 parts. The central component is a pentose sugar. A nitrogen-containing base is covalently linked to the 1' carbon on the sugar. This base can either be a purine with two fused rings, as in adenine and guanine, or a pyrimidine with a single ring, as
so at 1 dash position of the deoxyribose sugar, the nitrogen base, two types of nitrogen base can attach. They can be either purines that are double membrane ring chains, and pyrimidines that are single membrane chains like thymine or thymine and cytosine. And adenine are two types, purines are of two types, adenine and guanine. So in this way we get a nucleoside. We get a nucleoside where the deoxyribose sugar is there, and at 1 dash position any of either of the purine is attached or pyrimidine base is attached. This structure is nucleoside. But when at 5 dash position phosphate groups also attach, then this three component, when they are together attached, this becomes a nucleotide. So there is a difference between nucleoside and nucleotide. In nucleoside only two components are there: sugar attached with the base, and in nucleotide along with this two component, the third component also attached, called as phosphate group at the 5 dash position.
and cytosine and thymine. In RNA, the thymine base is replaced by uracil. A phosphate group is covalently attached to the 5' carbon of the sugar molecule. The phosphate group, the base, and the sugar make up a complete nucleotide. Note that the orientation of the sugar provides DNA with a 5' to 3' directionality. Base pairing only occurs between a pyrimidine and a purine. More specifically, a guanine only pairs with a cytosine, forming 3 hydrogen bonds indicated by the green circle, and an adenine only pairs with a thymine, forming 2 hydrogen bonds indicated by the red circle.
So this was the video. So I hope it is clear to you. So now we will see what is there in the NCRT. So this is the topic which is there after this diagram: primary and secondary metabolites. So primary metabolites are very simple. They are just the monomers which we have just studied. Sugar is a primary metabolite, glucose is a primary metabolite, starch is a primary metabolite. These are the things which will be directly used in the organism reactions, which will be directly either broken down or are synthesized. The organism is continuously synthesizing particular types of amino acids. The organism is continuously renewing its nucleic acids, making the new nucleic acids from the old one. So these the when the DNA will replicate when the cell wants to divide. For example, for suppose the cell requires the double DNA content. So double DNA content, how the cell will make? Cell will require the polynucleotides. Cell will require the nucleotide which we just have studied. Like the cell will require the sugar, cell will require nitrogen bases, cell will require the phosphate groups. So these groups or these compounds which are directly used by the organism, these are primary metabolites. Okay, but when this organism is producing some special things like alkaloids, flavonoids, rubber, essential oils, antibiotics, so when the organism is producing these things, we know that penicillin was the first antibiotic to be discovered by penicillium fungus. So this thing, this special component or special compound which is having a antibacterial property, penicillin, which is an antibiotic. So this special substance produced by the fungus is known as secondary metabolite. So those things Some of the plant also produce scents, gums, rubber. So these are the secondary metabolites, pigments. And here in the table, some secondary metabolites names are given, like alkaloid, morphine, codeine. These are used as medicines, drugs, for painkillers, and in the surgery. Terpenoids, monoterpene, diterpenes, essential oil, lemongrass oil, toxins, abrin, ricin, lectins, concavalin A, drug, vinblastin, curcumin. So these are polymeric substance, rubber, gum, cellulose. These are secondary metabolites. Now biomicromolecules and biomacromolecules. According to their size, according to their weight, if the molecular weight ranges from 18 to 800 Dalton, then we say that the substance is micromolecule, biomicromolecule. In this, the monomers will come. In this, the amino acid, sugars, these will come in biomicromolecules. But when the weight of the substance chemical compound is more than 10,000 Dalton, then it will become biomacromolecule. So, and these will be found in acid insoluble fraction. But along with the biomacromolecules like the polysaccharides, the nucleic acid, the proteins, we get lipids also in biomacromolecules, although they are not having such weight, molecular weight. So the reason is that they are not soluble in water. That's why they retain over the membrane or the filter. So they are separated in acid insoluble just because of their hydrophobicity. Now coming to the acid soluble pool, it is having the cytoplasmic composition and the membrane composition is acid insoluble thing. Now we come to the protein. Proteins are polypeptides. They are linear chain of amino acid linked by the peptide bond. Each protein is a polymer of amino acid, as there are 20 types of amino acid: alanine, cysteine, proline, tryptophan, lysine. Protein is a heteropolymer, not a homopolymer. So protein is having different kinds of amino acid. So different kinds of amino acid, when they bond with each other, make a long chain that becomes a protein. So protein is a polymer of amino acid. A homopolymer has only one type of monomer repeating n number of times, but here no protein is having only single amino acid repeated many times. So it is not homopolymer. They are generally heteropolymer. They are having different kinds of amino acid. This information about amino acid content is important, as later in your nutrition lessons, you will learn that certain amino acids are essential for your health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acid. Therefore, amino acids can be essential and non-essential. So, I told you earlier also, there are few amino acid, 8 amino acid, which are supplied to be supplied in the diet, and these are called essential amino acid, and other are non-essential. The later are those which our body can make. Our body can synthesize non-essential, while we get essential through our diet and food. Proteins carry out many functions. Some transport nutrients across cell membrane, some fight infections, and some are hormones, some are enzymes. So protein are having different functions. Here a small table is shown. You can see collagen is intracellular ground substance, trypsin is enzyme, insulin is hormone, antibodies fight infectious agents, and receptors, there are sensory receptors, smell, taste, hormone. These are also proteins. And GLUT4 is a membrane protein which help the transport of glucose into the cells. So GLUT4 is a membrane receptor of protein membrane receptor present in the cell membrane that transport glucose into the cell. Okay, so I hope this is clear to you, and we will study the next topic in the next class. Thank you. Please like and subscribe if you like the explanation.